Now we are going to part 2. So part 2 is going to be a little bit more less information, more observation kind of, right? I'm going back to the full screen mode. So now we did understand what mushrooms, where to find them, what do they grow on, how do they look. Now we'll narrow down to agaric type of mushrooms, which is the most thought after mushroom, imagined mushroom in the head. We'll look at its morphologies. We will understand what are the things to look at. We will understand by the end of it, even if you don't remember what to look at, you should know how to click a picture of a mushroom for a positive identification, right? So these are all the points which are going to be covered in this. And I will start now. Okay, so let's talk about this agaric type or the guild which are commonly found. And when we think of the word mushroom, these are the ones that generally pop in our imagination, right? So like we have nose, we have two eyes, we have two hands, we have two legs, we stand straight. Similarly, a mushroom has all these morphologies. It has a cap. It has some of them have decoration on top. Then there are gills on the underside, right? There are remnants of the partial veil. What it is, I will come. So some mushrooms have this, some mushrooms don't have this. So these are all the parts of a mushroom that we slowly break down into, and what are the kind of possibilities that you will see exist in these things. So the first part from the top of it, we okay. So this is a picture that you can click or take a screenshot of, or just click whatever. This is tips for mushroom identification, and this will make sense by the end of this presentation. Okay, so just give me a second. So this is how you click a mushroom. These are tips for mushroom identification. This is how you should send pictures for ID requests. Okay. Um, so coming to the shape of the cap, uh, these are certain morphologies that you get to see. And so it's very important that you click the morphological shape of this mushroom very clearly. They're cylindrical, they're conical, they're bell shaped, they're umbonate. You don't need to remember the names, but you need to first understand that so many shapes exist. And I have some examples for that. This is Coprinus comatus, commonly known as the ink cap mushroom. It literally forms into inks. You can see, like, when it is growing, it is very cylindrical in shape, right? So that is one. Then we have the conical cap. Conocybe is the name of this mushroom, and it literally looks like cones. So mushrooms do follow this stature. So when you click pictures, it's very important to understand what angle to click from and how do you click a mushroom picture, right? There are bell-shaped caps. Coprinellus section micaceous has bell-shaped caps. So you see, these three morphologies are so different. So one thing is that you really need to zoom in yourself to see what you're looking at. Then there are termitomyces mushrooms. I don't know how this pic got inverted. It was not like this before. So, yeah, invert your phone. That'll be easier. If you're on a laptop, it may not be easy. Then you need to turn your head. But there are mushrooms which have like a tip on top of them, like that. Then there is umbilicate cap. Umbilicate cap means it has a slight depression in the center. Now, I really want you to notice over here that this mushroom is growing on wood. Okay, if you can make out. Marasmius rotula over here is growing on wood. Now, I have a very similar mushroom picture which we discussed in the substrate part. Is this one? Is Marasmius, same genus but capillaris, which is growing on leaves. So a substrate is also very important while you take pictures of the cap morphology, right? So Marasmius rotula has like an umbilicate cap. There are convex cap of the agrocybe. Convex and bell shaped look very different when you start to see mushrooms. There is more depressed cap, so it's more depressed sounds wrong, but it's more deeper. The rasulas generally have this. This is a rasula species. So after the caps, we go a little bit lower. There are the margins of the cap, which is different again. There are smooth, there are crenate, striate caps, wavy caps. I'll show you examples. So there, this is a striate cap margin. If you just zoom into this, you can see there are lines over here. Okay, these lines are called striate cap margins. Leucocoprinus is a species which is really telling us, like, if you see striate cap margins, then it is something related to Leucocoprinus. There are more features you need to observe, but striate cap margins, Leucocoprinus have it. Some amanitas also have it. Some amanitas don't have it. Another example of Leucocoprinus cretaceous. So this was Leucocoprinus burnbaumi. Okay, this is Leucocoprinus cretaceous. You can see this has like nice lines at the edge of the cap, and they are very prominent lines. It's not कि दो तीन line है फिर थोड़े area में नहीं है. They are throughout. Then there are smooth cap margins where there is nothing on the edge of the cap. They're just smooth. Macrocype gigantea being one of the common edibles found in India also has smooth cap. Then there are wavy caps. Psilocybe cyanescens is one of the mushrooms which is a good example of wavy caps. So while it has a striate margin, the when it grows, it has like wavy caps also. So with striate margins, if you have a wavy cap, you can still say that it's something else. These are mostly point of observations. By this one observation, you will not reach to a conclusion of what mushroom it is. There are more things you need to observe. Then there is this beautiful mushroom called Panaeolus papilionaceus. And if you zoom into this, you will see it has like a beautiful petticoat structure. And that is why it is also called the petticoat motile gill. And uh, yeah, they grow on dungs too. This is soil dung mix. It's growing on. Then there are cracked caps. So when I was talking of termitomyces, this is one of the features of termitomyces that they have really cracked caps. Like they look like this. Again, don't just sit on one identification feature and call it termitomyces. I'm sure this is termitomyces after looking at other features also. But saying all this because this is a very tasty edible mushroom, but there are common lookalikes which may be poisonous. So uh, this is termitomyces, cracked cap example. Then there are hygrophanous caps. What does hygrophanous means? So these are all the same mushrooms, okay? But the amount of water present in each cap is giving you a different color. So this is a property which some mushrooms give and some mushrooms don't. This is called hygrophanous caps. Then certain mushrooms have a type of decoration on top of them, which is very different from other types of decoration. A lot of times after heavy rains, this decoration can go away. So you look at other features, but if they're not gone, it's very easy to clearly pinpoint on the genus and then the species level identification. So then certain mushrooms have part of the remnants of the universal whale. Okay. Some mushrooms have like they come from an egg-like structure. So for example, if in this picture you see this is the egg-like structure of this mushroom. Now as this cap will expand, this thing breaks down and spreads itself out like that. So I'll show you this is step 1. This is step 2 where it is elongating. You can see my pointer. This is step 2. And when it grows fully, it turns into something like this. This is Coprinopsis section. Coprinopsis observed in Bangalore, in Kaban Park, I think. So some mushrooms have remnants of this egg-like structure on top of their cap, which is again an identification characteristic. If you see an Amanita growing, Amanita mushroom, it also grows from sac-like, egg-like structure like this. Then, when there are colors of this egg-like structures, there are certain differences in the egg color also. Like you can see, this mushroom has like a darker patch of uh, decoration. So this is also an identification feature where the color of the remnants of the universal whale will tell you what kind of a mushroom it is. Again, point being that even if you don't remember everything, how to look at a mushroom and how close you need to get to rightly identify it is the main point of takeaway over here. um, while this mushroom has brown color caps, this one has the same colored brown color caps and brown color remnants. This mushroom over here has smaller remnants of the same size. Again, a very possible from the eye, but your eye needs to zoom in kind of. Coprinella section, orogranularity. Oreo I think is blue, and granularity is like granular, so like granular level of vellum. That's how it gets its name. And then we go from the edge of the cap and top of the cap, we go to under the cap where we observe the gills. So the underside is one of the most critical things for a mushroom because it has two things in it which are very important. First of all is how closely the gills are. There can be crowded gills, there can be closed gills, there can be subdistant or like distant gills. This is an aspero inocybe with beautiful purple looking gills, purplish shade. And then there are example of gills types. So Marasmiellus is one mushroom which has like distant gills, amazing distant gills it has. Then there are closed and crowded gills of the Gymnopilus. Then there are distant gills. So distant and subdistant, it comes through experience. What do you call it? Like you may call this subdistant also, but when you see a Marasmiellus, you will be like, oh, ये तो distant था, ये ज़्यादा है. Like this is way more distant. So subdistant gills are something like this, and then these are crowded gills of the agaricus. So underside of the mushroom is also very important. So you need to sometimes pick the mushroom and see. Sometimes what you can do is just use your selfie camera and invert it and look under a mushroom if you don't want to pick it. With gill arrangements, certain mushrooms will, if you see, observe over here, say this one, this is like one full gill from the edge of the cap to the stipe or the stem of the mushroom. Then this is not fully going in. And then this is one third of that also. So it's like one third, half, and then all in, right? Full. There are certain mushrooms which have all the gills which are going all the way from the edge of the cap to the stipe, and they are called equal gills. This is a rasula and it has equal gills. These are unequal gills, like I just said. It has like one small, bigger, then all the way going in, then small again. While rasulas have all gills going from this to this. So again, this is a very important observation feature. Then there are anastomose gills. Very beautiful picture of a lentinus over here. Anastomose gills are gills which are cross, like gills we generally see are running from edge of the cap to the stipe. Then there are gills which are running through them like this, like in a circular fashion. They're called anastomose gills. After gill arrangements, we go to gill attachment to stipe. Okay, and gill attachment to stipe is how the gills are connected to the stipe. Stipe is the stem. In mushroom language, it is called the stipe, right? So there are adnate, adnex, decurrent, subdecurrent, free, sinuate, emarginate, seceding gills, gill attachments. How do you check a gill attachment if you're not very sure? This is how you hold a mushroom. You slowly tear it, and then you'll come to this observation. So this is free gills over here because this gill and this stem has like a black line. And that black line is nothing but the shadow, which is saying that it is separate. So that is how you can check gill arrangement if you're not very sure. Um, Example of adnex gills from a mycena. You can see it is going, and then it is slightly attaching to the stipe. You can see pleurotus mushroom, which is the oyster mushroom. You can see gills going all the way down, like the stipe starts here. A lot of mushroom gills will end here. This gill will go all the way down till here, right? We can see that. So these are called decurrent gills. Then there are subdecurrent gills where it is going down, but it's not going all the way like oysters, but it's still going down. You can see this part where it's going down. It's called subdecurrent. Again, importance of observing a mushroom is more important. There are free gills. Free gills are like the gills which are not touching the mushroom's type only. Agaricus bisporus, the common button mushroom that we see, has free gills. Easier to see over here. This is a different mushroom. Obviously, this is a different mushroom. You can see the gills over here are attached. These are free gills. It's not attaching. So that is very important to observe when you're looking at a mushroom. Then certain mushrooms have different types of undersides. While most of them, these agarics will have gills. Some have teeth. Some have ridges. Some have pores. Pores like we have seen of bolids in the earlier part. This is hydnum and it has like teeth on the underside. So that's again a feature to observe. Undersides are observed only after bending or you pick the mushroom up. So a lot of people miss on that. These are ridges. So ridges are different than gills. A lot of people don't agree to this fact that they're different, but they certainly look very different. They are stronger in terms of their texture, and chanterelles are something which has ridges. Now, chanterelles are very commonly eaten in temperate climates. And why is it important to see this differences? Because there is something called the false chanterelle also, which is a common name of this species, the Hygrophorapsis. Over here, you can see gills, and over here you can see the harder ridges, right? So this one is Cantharellus or the chanterelle mushroom, and this is Hygrophorapsis or the false chanterelle mushroom, and they look very similar. Both of them have some different to different gills, and it can be confusing. Uh, Certain mushrooms have pores on the underside, and they are not its family. This is Philobolitus, and it comes in it is in Mycinaceae, which it's okay. It has spores, and these are glow in the dark. So one of the glow in the dark mushroom examples this is. After coming to from, we went from the cap, we checked the edge of the cap, we checked the underside gills. Now we talk about the stipe, which is the stem. So there are mushrooms which have like no hair on the stipe. There are mushrooms which will have little bit hair on the stipe. Okay. In fact, there are a lot of different type of stipes. You see every stipe over here is looking very different. Macrolepiota procera and chlorophyllum. These two mushrooms are very easily confused mushrooms. While chlorophyllum, if not cooked properly, is a poisonous mushroom, you can see this Macrolepiota procera over here has a snake type pattern, and this one over here is does not have any snake type pattern. So again, an observational difference. So stipes are very important to click pictures of also. Um, from this type, on this type, there are certain things like which is the annulus. It is present on some mushrooms, it's absent on some mushrooms. On some mushrooms, it's really beautiful. On some mushrooms, like it's smaller. This is like a beautiful skirt. So Leucocopra, I'll just zoom in a little bit so it's better to see. Leucocoprinus cretaceous has a stipe. Satharellas don't have stipe, like they don't have an annulus, sorry. Clarkeanda trichodes has a beautiful annulus over here. It's also called a skirt or the partial veil. Skirt is like in a slang type of a language. It's not officially called a skirt. And then Leucogaricus over here has a different looking annulus. Microsaliota has almost like a damaged annulus. Leucocoprinus fragilismus has an annulus. Now I'll show you another picture. I showed you this parasola over here. See, it doesn't have an annulus. And this mushroom look very similar sometimes. So the substrate is also like mostly soil. So when there is a soil mushroom which has like an annulus, it's called Leucocoprinus fragilismus, like this, or else it can be a Solotulosices. Then there are amanitas. Amanitas have some amanitas have a really nice partial veil or annulus. So from this type, we go to the base of it. Base of this type, some have egg structures, some have like a base which is really swollen like that. So that is also very important to observe. So again, when you click pictures, it's not to be so far away. It has to be zoomed in. You have to check the substrate out. You have to see if there's a vulva or the egg present or not. Right? This mushroom grew into this, by the way. It is a Volvopluteus gloriocephalus. You can see at the base there is like an egg, like there is an off structure which is not continuous. That is the egg. This is the egg. And this is the mushroom popping out of it. This is a base. It is not an egg. So you need to also clearly define if it has a base, has an egg, or it is just a swollen base. Certain mushrooms have a pseudorhiza, like the tomatomyces has, because it's coming all the way from a fungal colony, termite colony way in the ground. So that is tomatomyces. Odomancella radicata is also having a pseudorhiza. You can only find a pseudorhiza of a mushroom if you dig it properly and take it out and then observe it. So you can see the soil starts from here. It requires this much of digging. This one was almost this much in soil. Then certain mushrooms have a discus base. So the way they are attached to the wood or the substrate, it has a base. Discus base. This is another glow in the dark mushroom called Mycena chlorophyll. Really beautiful looking mushroom. Small they are. Some mushrooms have visible mycelium at the base, especially Coprinellus section domestica and Coprinellus section organellati. So this thing that you see over here is actually not moss, it's mycelium. And it's mycelium which is aged mycelium of this mushroom. And when you see something like this growing on wood, in fact, if you are somewhere in the ghats or if your house has a lot of humidity, you may find this mushroom growing inside your bathrooms or under your sink. And if you see this growth, you should be sure that this thing will pop up someday. And while you would not want mushrooms to be or fungus colonizing in your wood, only advantage over here you have is this mushroom is edible. So, but still get it ID'd. Yeah, free food. So after all these features from all the way top of the mushroom to the base of the mushroom, there are certain, like we went through all these characteristics from top to the base. Now there are other features of the mushroom which will tell us a little bit more about it. So that is by bruising and staining. So mushrooms like boletes, when bruised, they turn blue because of oxidation of certain chemical compounds in it. There are certain mushrooms in the Agaricus family and a very edible mushroom family, but if they are bruised and they turn yellow like this, they are not to be eaten because it's in the Xanthodermatia section, and that section is a poisonous section of Agaricus. Then there are psychoactive mushrooms which bruise blue because of the oxidation of psilocin, and psilocybin converting into psilocin becomes oxidized, turns blue. Magic mushrooms they are called, and they turn blue. So some people confuse boletes to be magic mushrooms because they turn blue, but boletes are oxidizing into a blue color because of a different chemical, and these ones are oxidizing into a blue color because of a different chemical. Then there are certain mushrooms whose gills you bruise or cut and they'll remain like release latex like substance, milky. So they are called lactarius, as the name suggests. And this is a beautiful mushroom called Lactarius indigos, which secretes out like a blue latex kind of a milky substance. It's my dream to look at this mushroom. I still haven't found it. Uh, yeah. So then after all this, there is the spore color. So a mushroom can be rightly identified once it has started releasing its spores. So it is quite when it's quite mature. So there are different spore colors also, like Paneolus, the motile gills we saw, or the ones like the magic mushrooms, which were was this example, this one. All these are pannivolus species from this one, and they have black colored gills. And if you observe them closely, they have this pattern. They are called mottled gills. Okay, Chlorophyllum molybdites over here is a poisonous mushroom if not cooked properly. Has green spores. Lacrimaria lacrimabunda is another example of a mushroom which has dark brown spores. So spore printing or spore color is an important feature. There are a lot of different spore colors. Volvopluteus has pink, dark pink spores like these. White spores of the Chlorophyllum brunnium is there. Uh, there is Entoloma species which are called pink gills because they have pink spore color. This is a very beautiful specimen observed from Wayanad. It's called falls in the subgenus Leptonia in Entolomaceae family. Um, Beautiful blue color with pink gills. This is one of my favorite specimens which I found. The purple to dark purple spores also. So it's very important what spore color it is. It really narrows down on the type of mushroom it is. There are orange spores of the gymnopilus. And then if we have time, we can go to this. Yeah, we can do 5 minutes, 6 minutes more. Awesome. So this was all about agaric type. We can also discuss a bit about polypores. Polypores are again these bracket type fungi which grow on trees generally or from the ground. They're called polypores because they are having multiple pores on the underside. I'll show you an underside picture, but a lot of polypores look so similar. So how do you identify a polypore? Polypores can sometimes be identified only by their undersides. And to prove that, this is the underside of a sterium. Okay, a sterium on top looks like this. If the pores are not visible, it is a sterium. And this is the underside of a tramites. Uh, Over here, there is one of them is a sterium, the other one is a tramites, but you can't really make out which one is which because till you don't go to the underside, this and this, you will not be able to make out which mushroom it is. So again, look below the mushroom, it gives you more information. Ganoderma has spores, but they are smaller. Reishi mushroom, this is the mushroom of immortality in the traditional Chinese medicine. Then there is microporous. I'll zoom in a little bit. You can see pores over here. They are visible to the naked eye. So then you can say it is a microporous. Yeah. Then there's Earliella scabrosa. So very random underside it has. Then there is the hexagonia. This mushroom gives that thing called trypophobia to people where so but I find it very cool. It has hexagonal structures. You can actually count 1, 2, 3, 4, 5, 6 sides, and that's why it's called hexagonia. This is just a comparative photo of a lot of polypose undersides. You can see they are beautiful undersides. Liela scabrosa, this being a type of Tramites betulina, Ganoderma. Then there are puff balls. Puff balls can only be identified through a cross section properly because then the amount of outer skin to the inner substance volume tells you a lot about the mushroom. Calvacia gigantea is a very commonly foraged mushroom for its taste and protein and micronutrient profile. There are certain stink horns which start with an egg, and when you cut them, you will figure out that it's a stink horn, it's not a puff ball. So, a cross section of a puff ball or egg like mushrooms is very important for identifying it correctly. You see, this is Scleroderma citrinum, and it has a really thick outer margin compared to, say, a Lycopodon over here. So, this thing is funny because in Karnataka, people eat this mushroom, whereas in USA, this is not considered edible, being the same mushroom. So we are pretty much at the end of the presentation. There are some common mistakes that people make, and through all the presentation done today, I just want to quickly rush through this edible mushrooms versus poisonous mushrooms and what we learned and how we can integrate in finding out how to look at a mushroom. This is Pleurotus austriatus and this is Omphalotus nidiformis over here. Okay, Pleurotus austriatus is edible, right? But they look so similar, right? Omphalotus are bioluminescent, by the way. Okay, all are not, some are. There are minor cap color differences, but they can be misleading. However, Omphalotus nidiformis will have a dark brown color patch on top of the cap. Okay, and so really get it identified properly before eating a mushroom. Okay, see, these are like common mistakes that people do, is what I wanted to show you. They are very similar looking, but still they are very different. Like, you won't find a patch like this on a lot of oyster mushrooms, but it's on Omphalotus. Then one biggest mistake that a lot of tribal people are still doing today is this thing, Macrolepiota procera, which is a very tasty edible versus a chlorophyllum molybdites. Now you see the cap is very same. It has a partial. It has like a nice long stipe. It has a nice long stipe. It has a partial veil. It also has a partial veil. Partial veils are different. This mushroom has white spore prints. This mushroom has a green spore print. Okay, then you can see snake skin pattern on this type. You will not see this on this type. So whatever we learned today, cap decorations can be similar, can be different. They have similarities, but then there are still differences. So you need to do the permutation combinations correctly first by observing them, then getting to a positive ID if you want to identify it yourself or just want to learn identification for that matter. Then there is Russula versus the Canthocybe. Russula is some species are edible in Russula, and Canthocybe is very poisonous. They both have these have decurrent gills, but from top they look the same. Russulas don't have decurrent gills. Canthocybes have velvety structures. Russulas don't have velvety structures. One of the biggest mistakes people do when they forage for these mushrooms, which sort of give you a psychedelic experience, a life changing experience, but they end up consuming the wrong mushroom, is Psilocybe cyanescens versus Galerina marginata. Uh, so these mushrooms grow on wood. Both of these mushrooms are saprophytes and grow on wood. Uh, while Galerina marginata, and they grow very nearby also, and they look so same. These are literally two different mushrooms. You can see that this mushroom will have like a brownish orange spore print. Psilocybes have a dark purple spore print or a black spore print if it is very dense. You can see the bottom of this type is a little bit black. This doesn't have like a black stripe, and obviously psilocybes will bruise blue and gallerinas don't bruise blue. But if you end up eating gallerina marginata, what may happen, these are called funeral bells, by the way. So instead of expecting an experience, you will probably want to rush to the hospital or you may die soon, and that is the fact of it. So while all of this is very important, it's very important to click the pictures of the mushroom nicely, understand what it is actually, and that pretty much gets us to the end of the session. Opening to question and answer, we have a decent 12 minutes for question and answer, I am guessing.
K. Ramanarayan has asked, thunder-related mushrooms in Uttarakhand usually refer to the morels or morchella species. Interesting to know of others.
Right. So while most of so in Uttarakhand, it's a more temperate climate where termitomyces don't grow and morels grow. Actually, mushrooms are associated with thunder, but if you go towards the Western Ghat belt or the other belts where morels don't grow, a lot of termitomyces grow after thunder mushrooms. So they are associated like thunder mushrooms. But the fact is that scientists say that nitrogen fixation is happening when thunder is striking. The other thing is mushrooms are fruit bodies, right, of the mycelium. So when the mycelium is injured through an electrical shock, it sends a shock in the entire network, and the shock tells them that okay, there is an issue. We quickly should make a fruiting body, and so mushroom pops out, and so thunder is sort of associated with mushroom growth. So it's not only termitomyces, it's not only morels. A lot of mushrooms which are probably coming out of soil will grow after a lightning storm. Yeah.
Okay. Then Pragna Yerabali has asked, I've heard that 90% of trees have mycorrhizal relationships.
Yeah.
Is that true? Maybe not mushroom fruiting fungi, but maybe the other kind.
Yeah, that's very true. In fact, there are this has been used as an application also where a lot of people who are into farming are not using chemical pesticides and insecticides and using fungus, because that fungal relationship with the plant and the fungi is helping the plant protect itself from other organisms, and these fungi do not form mushrooms. So yeah, a lot of these fungi which I gave example of in the start, where they help the plant roots, like plant associate themselves with and form roots, were also not macrofungi, they were microfungi, and there are a lot of microfungi which do this also. So it's true, it's not always mushrooms.
Okay. Then the leap Akoy has asked, why did some evolve to be edible and the others inedible?
So it is how our make is, right? So there are certain mushrooms which a monkey can have and we cannot have. There are certain mushrooms which the squirrels forage which we don't forage. So it is the chemical profile of this mushroom. It is not, so that is how we can actually see it and understand it better. It is not really about whether it is edible or not. It is what chemistry goes into your body, and if it is having a better chemical outcome which is good for you. There are a lot of mushrooms which are edible, which do not provide any benefit, and they are very not great in taste also. So that is another thing. So there is not exact answer of why did some mushrooms evolve to be edible or others inedible, but yeah, understanding that how that relationship works for us and the mushroom sort of tells us that there are some edible and some are not.
Okay. I've seen snails eat some mushrooms and some others have a small bite on them and then they're left alone. Does that give a clue about if they are edible for humans or not?
Amazing question. Yeah. So I will say snails will eat a lot of things that we humans will not eat. So, and then, so I will say that is not the correct one. Just said, I'm sure the question was posted before that, but squirrels will forage certain mushrooms which we cannot eat. Uh, Probably we can eat something which the squirrels cannot eat or someone else cannot eat. So, one of the things, while we are talking so much about edibility of a mushroom, I would like to get this to notice that mushroom cell walls are made up of chitin, and chitin is a polysaccharide, chemistry words, polysaccharide, which is not digestible in the human body. And because it's not digestible, it's very important to cook the mushroom properly, so the chitin breaks down and whatever is inside the mushroom as a nutrient profile is provided. Just some extra facts. Yeah, but no, if a snail is eating, still you can't eat it. You get it identified and then google the name and then find out about its human edibility. Yeah.
I heard it was phosphorus fixation. Hence, in the USA, they burn forest tracks to promote moral growth.
Phosphorus fixation. Yeah. So I am not really sure how much of a phosphorus fixation it is. They use ash. They burn forests in Himachal sometimes, which is not good to do, but that is to promote moral cultivation, because what happens is the mycelium is underground. The high pH ash is sort of helping the morels to grow, and it's also not promoting a lot bacterial growth and other micro or organisms which can be stronger in colonization compared to a macro fungi. So removing competition that way is promoting growth, and then selling morels in the market and the mafia rate is what they're doing. So yeah, probably that is what it is, kind of.
Then Safir has asked, please suggest any print or online material to help with mushroom ID.
Um, to help with mushroom ID, so I'll tell you how I learned mushroom identification, and maybe that will help, because this is mostly experience based. You always go out in the rainy season, you look at a mushroom, you get it ID'd on a Facebook community. Dude, Facebook has amazing people all over the world. There are scientists, there are scientists like me. uh, I'm not a scientist. I'm a law kind of a person who's got into a deeper science, but it is highly citizen mycology. So there are a lot of people who support this and are willing to help and share this knowledge, because this organism people don't know about the benefits of, and these people have helped me understand mushroom identification. Every different genus which is difficult to observe has its own sort of mushroom groups, where you can go to species level identification. So coming back to how do you learn about it, you can always say find two mushrooms a day, look at what it is growing on, observe its features, remember it, and then go into it. While something like this presentation or Safir can personally message me, I can suggest him some books. Books don't really help, because other than that, this presentation can just be given to you if you think it's useful, because it's like a compendium. It's like quick. But best thing is to learn yourself, look it up, Google it, read scientific papers if you don't find them to be very boring, and that will tell you a lot about mushroom morphology.
Then Radhika Kolgaonkar has asked if you will be doing any mushroom forays in Mumbai this season.
Yes, so thank you for asking that. I do mushroom forays mostly in the west zone, Mumbai being a center, and yes, you can DM me. My name is spelling is this. You can find me on Instagram and send me a DM, and I can tell you more about it. There will be mushroom forays happening in the next 2 weeks also in Mumbai, by the way.
Then Sohan Kogekar has actually sent a picture of a mushroom in the chat box if you want to have a look.
Correct.
He's probably asking for an ID, I assume.
So with Sohan has sent this, and
Actually, I've sent three pictures, and I was like, okay, after this whole thing that we have gone through with you, can we use certain things that you have told us and come narrow down to,
yep,
how to get an ID? Uh,
amazing. Amazing. So what we can understand from this is that this is a mushroom which is growing on wood, right? It is not an agaric. It is probably a polypore. But because we can't see the underside of it, we can't really narrow down to the idea of it. There's only one picture which is visible. Still, it
yeah.
can be a sterium. I'm sure this underside does not have
The
pores.
other two pictures I've sent, this mushroom I've never seen before, and I've sent the underside also. It is a polypore and it is growing on a tree.
Okay. It's a
It's
quite
red in
rotting.
color, if you can see.
Yeah,
It's
it's a
looking
quite
really
rotting specimen, I guess. It's a probably a Ganoderma which is rotting. I can't
okay.
zoom. Yeah. If you can just send me these pictures on somewhere else. What did you?
I'll send you somewhere else. It's fine. I got it.
Yeah.
Sort of.
Yeah, but it's a rotting specimen. If you have a fresher specimen, it will be much better. So I sometimes joke around this that a mushroom should only be identified when it has hit its puberty, because that's the best
Okay,
time to
cool.
ID it. Yeah,
Fair
to give
enough.
a safe ID. Yeah. Uh, which
Okay, then
mushroom
yeah,
fairy rings is
yeah.
Biplab asking that. A lot of soil growing mushrooms make fairy rings. You will find them in agaricus, like button mushrooms make fairy rings. In fact, yesterday I was at a friend's farm and found agaricus fairy ring. Fairy rings are just mycelial growth which is happening. Um, so it's see mycelium is from the center expanding in a circular fashion, right? And at some point it find a competitor in the soil. So maybe it won't be able to grow from there. So the mushrooms will just pop out from there, and it will look like it is going from a circular edge of that mycelial spread, and it looks like fairy rings. So that's how it gets its name. Thank you for asking that, Biplav.
I think we can take a couple of more questions.
Yeah, yeah,
Joel Alex has asked, at what point can we take the leap for consumption?
keep for
Um,
consumption. Cool.
is online ID groups, mushroom groups, if they are enough, or should we just stick to IDs from local person?
Okay, that's again a good question. What? Because yeah, in the end, if you want to ID it, and if you get into an issue, whom should you blame? I think I can also read that question that way. So see, there will be people like me who will give you an ID. There will be ID experts. There will be locals who will give you an ID. But in the end, it's your decision to eat it, right? And because I personally, I will say this because I don't want to be responsible for something that happens to you, and I would definitely do not want that. While I can provide a positive ID, eating it is completely on you. So yes, online ID groups will help you, locals will help you, tribals know this, but everyone makes a mistake. But that does not mean mushrooms cannot be eaten. I have been foraging mushrooms and eating them. So Joel, it's completely on you. But yes, you can trust people, and there are trusted identifiers. If you are on Reddit, you will see they have trusted identifier badge. If you are on a Facebook group, you will see a lot of comments from the same kind of people, and you can know that okay, this is a trusted identifier. If you are in a WhatsApp community, admins probably are the trusted identifiers. So that is how you can figure out, or you can ask the locals. And in the end, you can compare, you can Google, you can send pictures to multiple people, end up to a conclusion, and then you choose for yourself if you want to eat it or not. Yeah, that's the thing. Thank you, Joel, for the thumbs up. Yeah, what kind of mushroom is growing in South India, especially after Sohan sent this. So yeah, after that, next question is what kind of mushroom is growing in South India, especially after the monsoon rainfall? For example, the mushroom grows at root region of Cocos nucifera coconut tree. Is it good to trees or not, and what is the name of those mushrooms? um is this mushroom parasitic? Is this mushroom saprophyte? Is it growing through the root, or is it growing around the soil in the root? That is all that I need to first understand before I can even say or talk about or imagine the mushroom you're talking about, Bhuvaneshwaran P. Because um, and while parasitic mushrooms, if you are a farmer and if you have coconut trees, and if there is a parasitic mushroom, I'm sure it's not good for your trees, because it will probably take it over. And fungus is very intelligent to do that. But if it is a saprophyte, it's probably degrading the outer shell, the outer dead wood of the tree, which is not an issue. So please send pictures to me, and then we can narrow down and figure out if it is good or bad for
We'll
the tree.
just take two last questions because I think they're quite interesting, and then we can
Yeah,
stop. If anybody has any questions, if they want to get a mushroom identified, they can always get in touch with you.
sure, sure.
Um, so, do any mushrooms or fungi grow on sand next to seashores?
Yes, I have seen Coprinopsis growing on seashores. I have friends from Andaman. I have myself been to Maldives and I could see Coprinopsis mushrooms growing. So while I feel that it is not colonizing the sand, it is buried wood inside the sand, probably. In fact, cool fact is there are mushrooms that grow inside water also. There's something called Sathyrella aquatica, which is growing inside water, and it has fascinated scientists. And they're like, "Bhai, ये कैसे हो रहा है?" पर हो रहा है because they are very intelligent. So yeah, there are mushrooms which grow.
And and Mausumi has
Podaxis
asked, does the
pistillaris is one mushroom which grows in the desert areas. Sorry to cut you, Omkar. But
yeah,
Podaxis pistillaris is an edible mushroom which grows in desert. Amazing mushroom because it is growing in regions where you don't expect mushrooms to grow. So yeah, sand. If you associate sand with deserts and not seashores, sand there is also a possibility. Yeah.
Okay, so the last question Mausumi has asked, there is a common myth that mushrooms are sometimes considered non-veg. What is your opinion on that?
They're completely veg. In fact, all these vegan movements are asking you to eat mushrooms in spite of this. Um, I also mentioned in the presentation that when the Shravan Mahina is, I think it is going on right now, and this is the time when monsoon hits and monsoon gets a lot of mushrooms. So people stop eating non-veg, they stop eating chicken, fish, prawns, this, that, and they segue into eating mushrooms because they still provide you with the needed micronutrient and protein profile. So lot of myths around mushrooms. All mushrooms can be touched. White mushrooms are edible, red mushrooms are not edible is not it's not true. Identify your mushroom through understanding its looks. They are all not non-veg, and yeah, learn more about them because it's an amazing organism. Yeah.
Yeah, I think we'll stop here.
Okay.
So yeah, a big thank you to Vyom for such an engaging and eye-opening session. I'm pretty sure everyone must have enjoyed, considering the amount of questions that we got. And yeah, thank you to each and every one of you for being here with us today. We truly appreciate your time and curiosity. Just keep an eye out on our social media. Make sure that you follow us, follow Jim's Jungle Retreat on Instagram, because we will be announcing many more such exciting sessions very soon. And we look forward to having you there. Until then, thank you. Take care, and we hope to see you next time. Thank you.
Thank you so much.